The multi-billion-dollar anti-aging market includes products, supplements, services, and technologies geared towards slowing down the signs of aging. But beneath the surface, in the hypothalamus, a structure deep in the brain, there's a big breakthrough at Washington University's School of Medicine in St. Louis.
This is actually the first demonstration that manipulating certain specific set of neurons delays aging and extends lifespan in mammals.
Dr. Shin-ichiro Imai is a professor in the Department of Developmental Biology. Imai and his colleagues have had numerous discoveries over many years, leading to this latest one.
The hypothalamus, this tiny place within the brain, functions as the high-order control center of aging. So this time, our staff scientist and Kyohei Tokizane identified a very specific group of neurons in the brain. What we found is a very important feedback loop, the hypothalamus and adipose tissue, our fat. This circuit feedback loop regulates the pace of aging, process of aging, and lifespan.
In their study, the pathway controls signs of aging in mice.
Genetically manipulate to increase the activity of those neurons. And then this is the result.
Shown in green, key neurons producing an important protein called PPP1R17 in the dorsomedial hypothalamus of the brain activate a type of fat tissue to produce cellular fuel. This tissue is stored under the skin and in the abdominal area. This activated fat tissue releases an important protein, an enzyme called ENOM-PT. ENOM-PT returns to the hypothalamus and allows the brain to produce fuel for its functions. With age, the researchers found that the PPP1R17 protein tends to leave the nucleus of the neurons, and when that happens, the neurons in the hypothalamus send weaker signals, and that's when things begin to break down. The sympathetic nervous system wiring throughout the tissue retracts. The fat tissues no longer receive as many signals to release fatty acids and ENOM-PT. So with lower levels of ENOM-PT reaching the brain, the look and feel of aging becomes apparent. The research shows there's less energy to fuel the brain, along with fat accumulation and weight gain. The study shows what happens when these key neurons in the brain are activated in older mice, continuing the loop. The mice live longer than the control mice that have loops that lose the important proteins.
Really old ones, 30 to 31 month old, which is comparable to late 80s in humans. And if you look at these two mice, one is moving pretty actively.
Also, the look of aging is undeniable when comparing the mice.
You can clearly see the difference. And this one has a shinier, richer fur coat. This one has a kind of shabby fur coat. This guy has a kinked tail, inflexible. But this one has a very straight, still flexible tail.
The goal now is the development of new anti-aging therapies.
Still, and it's a kind of like a preclinical study stage. It's called an ENOM-PTB therapy, or ENET as an abbreviation. This is not the only one. This core feedback loop interacts with other feedback loops and then maintain, our body functions. Intervention to control the process of aging and lifespan.
So that one day, Imai says an average lifespan may be 100 to 120 years.
By understanding all of those feedback mechanisms, we could potentially come up with the very effective preventive methods, which let us avoid all the different kinds of disease and maintain the proper body functions. That's actually the goal of aging research.